I would like to thank our sponsors again, which is Milky Way Technologies Limited India, Agro Projects Poland, and Zhengzhou Industry China. They have been really supportive throughout the course of the event. And now, ladies and gentlemen, we move to a very special segment of this Day 1 of India Mushroom Summit 2023. We wish to invite Professor Sunil K. Srivastav, Department of Microbiology, Swami Shraddhanand College, University of Delhi, to join us on stage for the Golden Mushroom Oration Award speech.
Sir holds a PhD in virology and has completed a postdoctorate in Germany, Cambridge, and Tucson, Arizona. With an impressive tenure of over 4 decades in the field of teaching, Professor Srivastava brings a wealth of knowledge and experience to our gathering. In addition to this, he possesses years of research experience, underlining his commitment to advancing the frontiers of knowledge. Sir's area of research and specialization is both comprehensive and diverse, encompassing various domains such as molecular virology, cell biology, and many more. Sir has published extensively in prestigious journals of high impact factors like Nature Communications, PLOS Pathogens, Journal of Fungi, Journal of Clinical Nutrition, and many more. We are very pleased and grateful to have Sir here with us. Please give him a huge round of applause.
Good evening, everyone. Respected Professor Mago, Dr. Rekha, esteemed faculties, and dear students, this is an oration which I have designed specifically for the students. And Anurag asked me to make it a popular lecture event. So my lecture goes in a way that a layman can understand what I'm talking about. You don't have to be a mushroom specialist for this. And this is basically to motivate the students towards the fascinating world of mushrooms. And also, it's an idea from my side that what mushroom can offer to students as far as research is concerned, where exactly are we moving towards research, how this can help the Indian economy as such, and how it can help you to develop the skills, how the university can play a major role in this. So my abstract or my outline of the lecture has been published in the Mushroom Chronicles that you might have received. So without a further ado, I would like to start my lecture. Many of the things that I included have been talked about in the morning itself. So maybe I'll try to skip a few of them and then come to the main thrust areas, which are very, very novel and which has not been talked about, but has immense potential. And it is particularly for the students to take motivation from them. So as my oration says, advancing the mushroom path. As I talk about, I will talk about the milestones in mushroom sciences, what we have done and where are we presently. So mushrooms for long, we know that they are fungi, but many fungi earlier were being considered as plants. So it was not till 1969 that a new kingdom of fungi was erected. So when fungi used to be taught, it used to be taught in the botany department earlier, right? But as 1969, Roger Whittaker's classical paper in Science and subsequent papers established that there's a fifth kingdom, what we call as the Mycota. And this laid the foundation stone of a separate discipline altogether, and more emphasis was given onto fungi as a kingdom itself, and people started taking it very, very seriously or separate from the plants. As we go about, we'll see they are much different than the plants. They're not animals, for sure. Fungi are fungi. So they have their own identity, they have their own kingdom. So this is called the fungal kingdom. Then we had, a lot of talk about as to how many fungi are there when we are talking of a kingdom. It has to be very, very big compared to plants, animals. So the counting of fungi started, and one of the pioneers in this was David Hawksworth from the Commonwealth Mycological Institute in London, and he was the main person who is credited with working on the biodiversity of fungi. And these are some of his classical papers in mycological research. That was in 91, and then you see mycological research 2001, and again this is 2017. So this list goes on, and in every new paper in this review article, he would jack up the amount of fungi that he considered could be there on the planet. And as you can see, this is a beautiful world of fungi. Here only the mushrooms are highlighted. So fungi are both microscopic and macroscopic. And as we come to know about them, we find that they are more close to animals rather than plants. So this is an evolutionary tree. We'll talk about the phylogenetic tree and other things. So as you can see, the fungi are quite distant from the plants as far as evolution is concerned, and they're more closer to animals as such. So what are the advantages of having this? And just to let you know that the largest living organism on the earth is a fungus. So fungus as such are very, very small. Most of them are microscopic. Very few are macroscopic, what you see as mushrooms. That is one example. But the largest fungus is actually a whole network of a forest in the state of Oregon in western United States. And this finds a place in the Guinness Book of World Record. So this is a honey mushroom, that is Armillaria, as has been shown by the Guinness World Record certificate. And this is the humongous fungus, as it is called, and it's older than the Christianity and weighs about 400 tons. So it's a huge, huge network of almost the entire forest being covered by a single fungal species of Armillaria. So fungi as such are very, very big. They have a huge, huge network which you can't even imagine. And we'll talk about that. So I've already said, not all fungi are microscopic. Some are visible to the naked eye, and this is what we call as mushrooms. The whole 3 days thing we are just going to deliberate upon. So mushrooms are macrofungi that bear spores enclosed within a fruiting body, typically produced above ground on soil or on the surface of their food source. Mycology has taken a long course in the study in the University of Delhi since we are here. So I, along with two of my colleagues, we founded this microbiology course in the University of Delhi, and our college was the first to offer this. I was in fact doing my PhD when I was taken up for teaching virology. That was my PhD and postdoctoral specialization. So I remember Anurag, he was in my third batch. And when I used to teach them mycology used to be very, very extensive in those days. The whole Elixopolis we would cover in one paper. And the next paper would be all advances in mycology. We have reduced mycology to a very, very considerable extent, though it's not very good. But I remember the sparkle in the eyes of Anurag and many students who later on took to mycology. Anurag is one such example. So mushrooms are a microfungi that bear spores enclosed within a fruiting body, as you can see here, typically produced above the ground on soil, or even they can be under the surface of the soil. So both things you can see, and these are very big structures. So the fungal tree of life is very, very huge. What I've tried to show here is only the macrofungi through this paper of Nagy and Solzi, and this is an Advances in Genetics, and you can see the amount of thing, but this is another an outdated paper, 2017, and we are now in 2023. A lot of other things have advanced into this. So what I'm trying to tell you is every minute as I speak here, a lot of fungal diversity is getting added up, right? So what it used to be earlier in 2017 has exponentially gone up, because many of the fungi we are still discovering, and many of the mushrooms actually we are still discovering, particularly in the biodiversity hotspots. And just for your knowledge, earlier biodiversity hotspot used to be for plants and animals, but you would be knowing this, that fungi are also now included in biodiversity. So particularly in India, the northeastern states, which are really a gold mine of mushrooms because it's very humid out there, the temperature is very ambient for the mushrooms, and there are thousands and thousands of mushrooms which the world doesn't know even. And that area is wholly untouched. So much of biodiversity exists there, and it is for younger students like you to take up such things and let know the world what India has to offer. India has a very big geographical diversity, and so we have. So edible mushrooms we have been talking about since morning: cremini, morchella, shiitake, oyster, king oyster, lion's mane. I've just listed a few of them: white button mushroom, portobello. You just heard what are the advantages of these brown mushrooms, why they are more important, and so on and so forth. Ganoderma, and then straw mushrooms, Volvaceria volvuli, and then so many. So this is just a very, very few. This is just a tip of the iceberg, and what we really are using as edible. So there are so many which we can use as edible, but we haven't yet explored them. The classification of fungi goes back to Linnaeus' time, where Carl Linnaeus, you must have studied this. This I teach in the first year of my microbiology student in introductory microbiology, and this is the taxonomy is very, very important, because this is very, very confusing as far as the mycologists are concerned. So now we have tree of life, archaea, bacteria, and eukarya, and fungi are in eukarya, so they have all eukaryotic characters. And most of the fungi actually don't make mushrooms. I've already overemphasized that. About 90% of them don't make. This is Saccharomyces cerevisiae, which you are very familiar with: yeast, rusts, chytrids, molds, smuts. So most of these fungi are microscopic, but we have a whole range where you are getting macroscopic, and this is the most familiar thing. Right from the school we are taught about what a mushroom looks like, but this is not the only mushroom or the type of mushroom. So gilled mushrooms are taught in the curriculum right from the school stage. So everybody is familiar with this. So this is one very, very small representative of a mushroom where you have a button stage, and the button stage grows. Is it the pointer? Okay. So this is the button stage, and it goes, develops a mycelium and develops into a stipe, and then this whole mushroom structure. If you see, these are the gills, and under the gills you have the hymenium, subhymenium, and wherein you have the real spores, the fertile layer, and these basidiospores get released, and the cycle gets completed. So mushroom is just a part of the organism. As I said, many of the things you are seeing are mostly underground, and some of the things we are seeing which are above the ground, and this is how the spores get released. This is the time-lapse photography, which I'm not playing that because otherwise my lecture will go very, very long. I've just taken a pic of this, and then the hyphae, which is 1/6 of a human hair, just to put things in perspective, it's so thin. It's 1/6 of a human hair, and this forms a whole network, as you can see, and it grows underground. And then when the conditions are productive, what we call as the pinning, the primordial things comes out, what we call as a button in case of Agaricus bisporus, and this then fruits into a full structure of mushroom. So this is the mycelial network, which is very, very thin. This is very important because if you dig the forest floor, you will find a huge network. And this is all helping the forest to survive. Without this, even the forest can't sustain itself, you know this. It's so important. So this mycelial network, this fungal whole kingdom is so important just even for our very existence on this planet. So I just wanted to emphasize on this. So this, I'm just trying to tell you what exactly is the cycle, because this lecture is made for a person who even doesn't know mycology. And we are going to face these challenges in national education program, where we have to tell students who don't have any background knowledge. So this is how the whole mycelium looks like. You have septum here, and because these are higher fungi, both basidiomycetes and ascomycetes, they have septa. And then this is a very, very important term, Spitzenkörper. Spitzenkörper in Deutsch. I studied Deutsch for 3 years. So this is a very, Spitzen actually in Deutsch means pointed. So you have a pointed body. And this pointed body keeps on advancing, you know. And I'm just trying to show you how this advances and how this physiological layout, how this lifestyle is helping us. I will try to bring out that point. So Spitzenkörper is very, very important. This pointed body keeps moving, and as you can see, it's just putting out these red dots. Can you see in the front? So these are some of the enzymes that are getting secreted. So this uses a battery of enzymes which are produced in these vesicles, again the red dots, and these come out of the hyphae, the growing tip, and release, and whatever complex material is there, it gets decomposed. And this is then absorbed. So it has an absorptive mode of nutrition wherein it would throw up the enzymes outside. You give anything to the fungus, it would try to digest it. So that's very good actually. And so you can have an in vitro culture, you can throw them into the petri dish, and you will get a metabolite as a result of the metabolism of that particular fungus. So this nature has given to us, and I'm just trying to explain why this is so significant. So this is the Spitzenkörper, and as you can see, the fungus hyphae has advanced a little. It deposits enzymes in the vicinity, add membranes to the hyphal tip, create a space ahead of the growing hyphae, because it is advancing, it is just moving like this, right? So this is what happens actually in a petri plate when you're growing a fungus. So from the center where you put the culture, it will radiate in all the directions in a petri plate, and the whole thing would become finally like that, colonized, right? So this is the haploid and dikaryotic. Now, this is very, very important. Why it is important, I will just let you know. It is taught to you, but many people don't understand what is real importance of this. So there are two nuclei. Can you see? One is red and one is blue. So there are two mycelium, and they have to be opposite mating types. Once they come together, there's a fusion. Can you see this part? The hyphae has anastomosed together, and the nuclei have come together. However, they haven't fused. So it's only the hyphae strand that has fused, but the nuclei haven't fused as yet. So these two nuclei, as is showing in my pointer, remains the same. They don't fuse, right? So this is therefore called a dikaryotic mycelium, because there's a dikaryon. Karyon means nucleus, and there's a dikaryotic mycelium that is developing, and this dikaryotic mycelium goes to produce the fruiting body. And within that fruiting body, what happens, you get structures. If it is a basidiomycete, if it's an ascomycete ascus, basidium is formed. So these club-shaped structures that you're seeing here, these are the basidium, and these are the sites where this would fuse. These two nuclei, these dikaryotic nuclei, would come and really fuse. It's like your egg and sperm fertilization, which we have in our body. So this takes place, and immediately you have meiosis, and immediately you get 4 new haploid spores. So the diploid phase is very, very small. Mostly it is haploid, or it is dikaryotic. These are the two phases that the fungi normally knows. But for a very, very short period of time within the basidium, you get the diploid stage. Why this is important? Now let me explain. This is important because the whole genetics is here. And if you want to make any improvement in the strain of a fungus, you have to understand this genetics very, very nicely, because you can manipulate the haploid. You can manipulate the haploid because anything haploid is more easily amenable or manageable genetically compared to diploid. So you can always have a control of which haploid spore you are taking and which you can mate with another haploid spore and get the desired diploid which you want. So you can combine the characters. That is what I want, and it's so easy to do. It's existing right in the Petri plate. It's existing in the basidium. You have to just do this in vitro. It's so easy. So at the end of the talk, you would get convinced that mushrooms are the best model organism that the nature has given to us. You want to have any dream, it can be fulfilled through mushroom studies. It is as simple as that. Okay? So these are the gills. You can see these knife-like structures if you upside down the undersurface. And this is what is happening. These gills are there, and they're lined with these basidia, which I just talked to you about. And these are having at the sterigmata, the tips, these basidiospores. And these basidiospores would get dispersed and go to another place, and the whole mushroom cycle keeps on going. Okay, so the next is the diversity of mushrooms is very, very huge. As you can see, most of them are gilled mushrooms, which we commonly see. We have bolete, you have polypores, you have cup fungi, you have morels, you have toothed mushroom, puffball, earth stars, clubs, corals, stinkhorns, jellies, crusts, and steroid fungi. All these are mushrooms, right? So these are the gilled mushrooms which you commonly see, but this is not the all. Many of them are edible, which we have mostly used, but many are deadly poisonous. Many of them are highly, very, very colorful, and that's the another deceptive thing. Boletes, these have also capstock like the gilled mushroom, but instead of gills, boletes have a tightly packed tubes, you know, and packed tubes to increase the spore producing surface area. The idea is to have more surface area because you want to have more spore production. More spore production means more amount of dispersal and the survival of the fungus. So the whole mushroom is soft and fleshy. Tubes can be separate from cap. Boletes, many of them are mycorrhizal. I think most of you would know mycorrhizal. Mycorrhiza are association with higher plants. This is a very, very ancient relationship, which is so important for our existence, so important for now people have exploited it for agriculture, but the whole sustainability or the diversity is dependent on this relationship, whether it is endo, whether it is ecto, and all kinds of relationship. So boletes form mostly the ectoparasites, ectomycorrhiza, sorry, and then the polypores. Here you have these pore-like structures, and these are also producing the spores within them. Then the chantilliers, you can see these structures, just trying to, they also form mycorrhizal species. Then the toothed mushroom, here is the lion's mane, which is very, very important medicinally, Hericium erinaceus, and then depressed hedgehog. These are very, very important species. Again, there's puffballs, earth stars, might have seen. So they're beautiful structures to look at and how this dispersal of spores takes place, clubs and corals. So coral fungi are very, very important again. And then the stinkhorns, which produces a very offensive odor. Even some species can be eaten in the egg stage. So this is also, jellies are one. Jellies, you can dry them and then soak in water. They will again retain their shape and the entire thing. So Auricularia is one such example of this. Then the cup, saddles, morels, and other vascomycetes, wherein you don't have an umbrella-like thing. Like the cup cap, we have a cup-like structure. And within the cup, you have the hymenium within the cup, right? So this is the cup, and here you are finding the layer of the hymenium surrounding inside, and this is the ascus with the ascospores. They are very highly prized, I think, you know. This is something new to India, or we were having, but we didn't started cultivating it. Yes, but it's now very, very important and very lucrative industry. And these cups, like orange peel fungus, and then the saddle fungus, and morels. Morels actually are in between the two. It's not a sessile structure like this one, but it's a stalked structure like this, as you can see. And there are not one cup, as we see here, but there are multiple cups. The whole fruiting body has multiple cups in them, and within them you have the whole ascospores, right? Then cordyceps is also one ascomycete fungus, which is very, very important. Again, very, very medicinal, important properties, which you must have studied. The mushrooms on the substrates that they grow are very, very diverse: soil, wood, even dung. Psilocybe, I think you know, grows on dung. It's a coprophilous fungus. So now mycorrhiza is very, very important, and many of the mushrooms, in fact, form association of mycorrhizas. I just wanted to tell you the whole industry, the forestry, has now taken mycorrhiza as a very serious subject, and they are inoculating all the pines, oaks, and all those things. They are putting in the mycorrhizal, ectomycorrhizal culture before they put it into the thing to increase productivity, because mycorrhiza trade nutrients with the host. It provides those nutrients which the plant cannot take on itself, and the whole network, and they try to get this. Ecological roles of fungi cannot be overemphasized. It's very, very important. Again, the whole forest floor. This is one of the figures from the forest thing. There you can see how, so the degradation of the forest wood, you know, the cellulose, hemicellulose, and the lignin. So we have white rot fungi, brown rot fungi, I think you know this, where everything gets digested, that's a white rot, and brown rot is still you have lignin. So this all is very important, and this, why I'm stopping on this particular slide, is very, very important, because when we are trying to make biofuels, your country is really aiming at producing the biofuel. It wants to be a world leader in biofuels. These fungi are again very, very important, because the white rot and the brown rot, because lignin is a big problem, and lignin you can degrade by the fungi in this particular group. So identification of mushrooms has always been a very, very big challenge, right from days when we started foraying for mushrooms and trying to identify them. It's very, very important whether it's poisonous, whether it is non-poisonous, whether it should be eaten, and how do we identify. We were doing many things, like you just cap out this cap and put it there. You can put it either on a paper which is half black, half white. So if you have colored spores or the white spores, it will show up on black, and if you have black spores, it will show on white paper. People also use glass, plexiglasses, and you get a spore print. So spore print is a very, very important technique in identifying mushrooms. And of course, the spore colors is one other important thing. So identification has been a challenge, and I'm just trying to skip those slides. Yeah, I'm getting already that, yes, I can understand that. So the diversity is one thing, but now we are in the nucleic acid age. So nucleic acid is the most important thing, whether it is what kind of DNA, what is the genome of the fungi, and everything is now coming to genome. Carl Weiss, a microbiologist at the University of Illinois, he changed the whole thing when he gave the Tree of Life in this important paper in PNS, and he gave the domains, what we have, the 3-domain classification, and this is, so I'm just rushing through my slides because the time given to me is very, very small. So this is the phylogenetic tree of life, and as we see, fungi are much closer to animals than to plants. And the tools that we use is mostly the ribosomal RNA genes. So here are the two groups, the basidiomycetes and the ascomycetes. Dikarya, I have already explained why they are so, are the ones. And this is the ITS regions, you know, internally transcribed sequences in the ribosomal DNA, which is amplified through these primers. And this is now used as a taxonomic marker as to identify which fungal species we are talking about. So from the way we used to identify mushrooms, we have come a long way, and by DNA barcoding. So every mushroom has a DNA barcode now, and this is now in the database. So like you have in the grocery store, So which mushroom is this? You can just have the barcode and know what mushroom you are dealing with. So these are the MRC guidelines. I don't have the time to go through this. So these are all molecular, morphology, ecology based. And this is the phylogenetic reconstruction. The phylogenetic trees are very, very important. Penicillium chrysogenum, I think you know, Fleming's original thing has now changed to Penicillium rubens, what used to be earlier. And these are some of the products that fungi can produce. And one example is shiitake mushroom. So I don't have the time again to go into this, but you have been hearing since morning that there are many, many uses of the metabolites of the fungi. MSA, multiple sequence alignment, is very, very important. And now we have a whole set of metabolites, the proteins, and we are trying to annotate the genes because we have a whole genomics data available with us in the NCBI and many, many databases. And we are trying to see which fungi is producing what protein and why, how this protein can be used. A crystal structure of that can be made and try to exploit it for medicinal properties and industrial properties and things of those kinds. So MSA. So fungi for food, we have been emphasizing this. And these are some of the nutritional values, again, which you have been hearing since morning. I don't have to repeat this. So it has immense benefits which no single food source can do. It has all the vitamins, it has all the essential vitamins, the amino acids, and so many things, and medicinal properties, immunomodulatory properties, selenium, vitamin D, which are so many talked about things in health benefits these days. They are present in that single humble mushroom, right? So it's a very important product. This I had tried to make how the fungi mushrooms are grown. This starts from a culture, and this culture in the HEPA filter front, you just put that, subculture it into Petri plates. You can do it in broth also. And then finally you can put it into some substrate, what we produce, the spawn and the grains, and this whole thing becomes fully, and then you can do secondary, this thing transfers, and thousands of transfers, and this spawn culture can be shipped to various places. Even you can have these kinds of pellets which you can drill holes in the logs. This is for shiitake, and this is how we grow this. So this is the substrate in which we grow. So this all. So coming to the main point, what I want to emphasize through this lecture is mushrooms should be a very, very important part of our curriculum, because India is going to become a global leader, and we cannot be far behind if we are not trained into mushroom. And India has such a huge diversity, biodiversity, so many species which we still haven't explored. And we have such a manpower where we are training so many students. So this is one such figure that has to be in every curriculum. It has to be taught how to grow mushrooms. And your principal is doing a wonderful, marvelous job in spearheading this skill development program in the university, because this is very important for a vocational training, not only the formal education that we have, but also vocational part of the curriculum. So most of the people should be taught exactly how this goes about. These are some of the mushrooms in India. These are very, very few, unfortunately, but we are growing these many only. We have to expand this, seeing the world market. And this is some of the Indian regions. I don't have the time again, what I wanted to show here. Let's just skip this. Global mushroom production in the world. Dr. Manjeet Singh was just talking a little while ago about the figures, whether they should be relied upon or FAO and other things. But anyway, China has a huge chunk. Some say 75, some say 90% of the world's market. India has a minuscule, just hardly 2%. And we have to expand this. We have to go a big way. And this is why the advantages of India has 2% of the world's mushroom production. China accounts, okay, I do not know how reliable this data is, but India has cheap labor, India has very well-trained manpower, well-educated teachers, who can take up this task and do this kind of a thing. Skill development can't be overemphasized, economic empowerment to most of our population, increase employability, nutritional security, and of course career advancement. India fights what is called mushroom protein malnutrition, because most of the people are in India deficient in protein, and mushrooms can be a very, very good source of that. Medicinal properties, again, Ganoderma, so many of them, Cordyceps sinensis militaris. So golden oyster, you just found some of the mushrooms that we talked about, the king oyster mushrooms, reishi, shiitake. Then reishi has many medicinal properties. Again, I don't have to even used in cancer chemotherapy. And then turkey tail, then this is even lion's mane is used in Alzheimer's, dementia, and things. So those diseases, medicine. So these are health benefits. They're innumerable. You can read on the internet and find that. So these are mushrooms and nutraceuticals. Just before me, a lady was talking to you about, one professor from Kerala. She was talking about the nutraceuticals. Again, she has talked about, so I don't want to emphasize it. But magical mushrooms, this is something very, very new. At least we are not doing it in India, but many countries have now standardized this thing. They have legalized the use of psilocybe. So psilocybe is this mushroom, and if you take the stipe and press it, you will find a blue region that shows it's a psilocybe. This is how it looks like, the fruiting body. And psilocybin, once it goes into our liver, the hepatic system converts into psilocin. And if you were to see psilocin structure, it resembles your neurotransmitter, what is called serotonin. So serotonin is a very important neurotransmitter. It's being prescribed for many, many neurodegenerative disorders, depression, anxiety, and this is now legalized in many countries and people are using this. So another important thing is antimicrobial resistance. We are fighting a pandemic of antimicrobial resistance, and this mushroom system can be used, and it's very, very unfortunate, as I'm trying to show through this, is from 1943 when the first penicillin was discovered till 2010 we could discover a few antibiotics. It's so unfortunate. From 2010 we haven't discovered any antibiotic, and all the antibiotics that we have become bugs have resistance. Mushrooms can offer a very important platform, right? There's so many studies. I don't have the time to go into this, but just one example I'm taking. Here is shiitake mushroom. You can see this particular shiitake culture, and this is the 2 E. coli. And what happens later on is, the slide I have not put, so the whole thing gets inhibited, the inhibitory zone. So there are so many mushrooms, you will hear about them, that they will be used for antibiotic production. Biofuels, I've already discussed. Fibers, textiles, packaging materials, building materials. India has a huge biodiversity, and Northeast India is one such thing. Nanoparticle production through mushrooms is again a very, very promising area, wherein, because it's generally regarded as safe, it's a grass species, you know. And for industry, the grass species are very, very important, and mushrooms are all grass species, and they can be used very nicely for the nanomaterials that we use. I'll take 1 minute now. These are my closing slides. So we are fighting pandemic, and Ebola pandemic had been there. SARS-CoV-2 came and everybody forgot about Ebola. So these were so many pandemics that we had in Ebola. When Ebola outbreak for the first time struck West Africa in 2018, what happened? The monoclonal antibodies were used. We did not have any licensed monoclonal antibody to be used against Ebola. And there was a lab in the US, Charles Anson's lab, and he was producing these monoclonal antibodies in plants. Now the biggest challenge that that person had, that the glycosylation of plants is much different than that of animals, right? So, but this monoclonal antibody that was produced in plant got an emergency license by the President of the United States, Barack Obama, He licensed it. And he could save the US citizens, the US physicians, the US nurses which were deployed in the West Africa. Their lives were saved. And subsequently, many African patients were also treated with this. The challenge in Anand's paper and the company that says is the glycosylation. Now, mark my words, mushrooms glycosylation is much, much more similar to human beings. So if we were to produce the monoclonals in mushrooms, they will be much better compared to the plants. And the types of rejections that we get in plants is, so we already have a case study where the lives could be saved against a deadly disease like Ebola, which is a death sentence. So lives can be saved. So there's a lot of work in India. We are not doing this, but yes, this is one which I wanted to tell the students. I'm trying to tell you, so this is the SARS-CoV-2, and one such example is mRNA vaccine. mRNA vaccine has been a life savior. You know, the Pfizer-BioNTech and the Moderna vaccines in the United States produced this, yes. And these 2 Nobel Prizes were given in 23 this year only on messenger RNA vaccine. And mRNA vaccines we can produce in mushrooms, and it can have all the codes changed. Then we have the checkpoint inhibitor therapy, wherein you use antibodies as shown above, and these can be used against the checkpoints. I don't have the time to explain this, though I would have loved to do this. So these are the antibodies which are all commercialized. They are FDA approved and they can be used, but their price is very, very high. If we start producing them in mushrooms, the price will go down, and it's very, very important for countries like India, the low middle income countries, and Africa to reduce the cost. So a person who has cancer cannot be given because of the high cost in India. No insurance company covers that. But you start producing this in mushrooms, I think no cancer patient will be deprived of this. So this is some of the messages. These were the 2 gentlemen who did this. And then we have CAR T cell therapy. CRISPR-Cas9 is a very revolutionary technology wherein you can edit any gene. And the best model organism to do this is mushrooms right now, wherein this can be done in Petri plates, this can be done in test tubes, the easiest thing that we have. And this got a Nobel Prize. And with that, I will thank you, and thank you for your kind attention. And if you have any questions whatsoever, I would be more than happy to answer.
I'm really happy that you mentioned that it needs to be incorporated into curriculum and
Sure.
that it has to be more advancedly researched, studied. But I want to ask specifically that you focused a lot more on the science of mushrooms and their capabilities and what we have not discovered about them, and the research that can be done to unlock their properties. But unlocking their properties when we are only at 2% of the global distribution supply of mushrooms becomes a very difficult prospect to invest in research and development, right? So how would you
Why is it difficult? Because if you give the industry skill trained, industry ready students who can employ them, they don't need any training, and they can straight away start working. They have the educational background. They are not technicians. They have been educated in this. That's why I'm emphasizing on the curriculum. You're not making robots like in China, right? You are a country where the science is so immense. The faculty is so educated. You know each and everything about mushroom cultivation. That is why I was complimenting ma'am for this. Yeah, because skill, not only you don't have to get admission into university. You can be just a vocational this thing and you can get trained. Right? So if we give the industry ready students, right? And you can catch them young and train them.
So you're suggesting that it is a simpler venture to explore, employ students and get them to explore this area in a lab?
It's not forcing the students. You just train them. It is for the students to decide. They have the freedom whether they want to pursue it or not. But industry has someone who is trained. Right?
Thank you. Please wait. Sir, please go. Thank you so much, sir. Now I would like to invite Dr. Preeti Verma to felicitate, sir.